everybody. My name is Nick Tucker. I'm an associate professor in the School of Engineering at the University of Lincoln, and I'm going to tell you the story of the development of a COVID-specific mask that I did with colleagues in the School of Pharmacy and the School of Design. And you can see my heading there, Materials Research and Development in the Time of COVID. As practically anyone who was able to jumped in to try and help out. And my subtitle there is The Slaying of a Beautiful Hypothesis by an Ugly Fact, which is a quote from Thomas Henry Huxley, who was Darwin's bulldog. And you'll see the problems that arise when you try and take something from a gleam in the researcher's eye to a useful product. That's the nature of this story. So our contents, we're going to look at nanotech in the beginning, and then electrospinning, which is my great love in terms of research, and how this leads into filters and how it could be developed and scaled up into a highly effective COVID-proof mask element. And then we'll review what we actually finally ended up with when our lovely hypothesis came in contact with horrid reality. So nanotech, the notion was first proposed by Richard Feynman back in 1958. So December the 29th, I was 4 months old when he proposed this. And what he said was, why can't we directly manipulate matter at the atomic scale? And this was an idea that caught on. If you want to read his original lecture, it's actually available as a download. It's quite interesting. And he was a bit of a card. He was a noted lock picker and also very good at playing the bongos. We digress. Let's have a look at electrospinning. So this is intrinsically a very simple process. That what we do is we take a strand of liquid polymer. Now that could either be a solution of polymer, a dissolved polymer, or it could be a melt, and we stretch it out until it reaches the nanoscale. And the apparatus we can see here, quite simple, because all we're going to do is we're going to subject it to an electrostatic field. So exactly like you would do when you rub a comb on your sleeve and pick up bits of tissue paper, that's actually a very strong force. And what it can enable us to do is we can take a drop of polymer, and over about 150 millimeter distance, we can accelerate it from 0 to about, let's say, 70 meters a second. And it consequently gets a lot thinner, and it deposits into a random mat. Our little picture there actually shows a non-random mat. We've diffracted a laser through it. That's just me showing off. So if we had a meter squared map of this mat of this material, so a fiber, let's call it a nanofiber, a diameter of about 100 nanometers, and we deposited it about 10 microns, 10 millionths of a meter thick. The surface area of material we'd have on that square meter mat would be about 40 square meters. So we've got a terrifically high surface to volume ratio with our material. And also note that might actually be a single piece of fiber. Let's look at a schematic of the process. So the spinning tip is just some convenient way where we can produce a drop of material, a little droplet, a bead of our liquid, which, as I say, may be either a molten polymer or a solution of polymer. What we need to make this work is we need enough molecular cohesion in that liquid so that when we subject it to an electrostatic field, so this spinning tip will be maybe held at, let's say, 15 kilovolts, and our target 150 millimeters away will be earthed. So there's a strong electrostatic field there. And what it does is it pulls the little bead of liquid at the end of our spinneret out into a thing that looks like either one of the domes on the Brighton Pavilion or the Kremlin, whichever you prefer. And as our attractive force overcomes surface tension, it shoots out a jet of liquid, very thin jet of liquid, which is accelerating rapidly. At some point, we don't really know where, the solvent will have evaporated or the melt will have frozen, and this will become a solid fibre. And if we could stop here, that would be great, but the flight path isn't simple. What we believe happens is as it accelerates, it hits its terminal velocity, so there's a little bit of a compressive force there, and that's enough to buckle the fibre and produce this expanding helix. So why is it expanding? It's expanding because it's got a massively high electrostatic charge and it's mutually repelling itself, and then it whacks into the target and is stuck there and deposited, and most of that charge leaks away to Earth. Now, with a bit of luck, I can persuade a video to work that actually shows this process happening. So what we could see there is we could see this is our spinneret, and a jet of liquid is being drawn from it, and at some point here the flight path converts into this intriguing expanded helix. And by the time we've got to here, there's not much point in showing anything further on, because it's dropped below, in terms of diameter, the wavelength of light, and we can't see it. So wavelength of light, let's say green light, about 400 nanometers. We are handsomely below that. And this video was taken at 10,000 frames per second. It took my student John 3 months to get it all sorted out. It's quite a nice piece of work. So, in summary, all we need for electrospinning is we need my laser pointer to work again. Okay, we need a high voltage source, we need something we can spin, and ideally, this is many academics are striving to do this even as we speak. We need an understanding of the theoretical principles of the process in order to control it properly. Now, here's a couple of scanning electron microscopes. This great big knobbly tree trunk thing here is a bit of spider's web, and here are some nanofibers deposited over it. This is a scanning electron microscope picture purely taken to show the scale of the fibers. And sometimes the way the fiber draws out, we get some quite interesting morphologies. So this is all one fiber, and due to inconsistencies in the mixture, some of it is drawn out thin and some of it not. This is just a fiber drawing thing. It's not specific to electrostatic control. We'll leave it there. Why on earth would we bother to do this? Here's a little bit of a case study, one that I did earlier, working in New Zealand with the New Zealand Institute for Plant and Food Research. And that rather strange looking fish is known in New Zealand as hoki. It's also known as the blue grenadier. It's a pelagic fish, it's an ocean fish. If you're keen on the Linnaean business, it is Macaronus novo-zelandii, which I suppose gives us a clue that it's from New Zealand. And it's a sustainable origin fish. They catch quite a lot of it. I believe if you are buying a random white fish fillet in Tesco's, very likely it's hoki. It's quite a nice fish to eat. Of the fish, 65% of it is giblets. It's the skin, it's the liver, it's the bowels, it's the head, it's the bones, it's the fins, it's the stuff we can't really normally eat. And the fillet is about 35%. So if we look at the prices, we've got prices in pounds going up the side. So these are price per kilo. So that's 65%. If we just send it for fish meal, so it can go as fertilizer or it can go as bone meal for the garden, fish meal for the garden, we're looking at about 50p a kilo. The fillet is going at about 6.6 pounds a kilo. If you took the liver of that unlucky creature and extracted the omega-3 oil, when you went to the chemist to buy your odor-free omega-3 capsules, you would be paying about £66 a kilo for that fish oil. Now, my colleagues at Plant and Food extract this material. It's used as a fining for wine and beer to take the haze out of those products. And they're selling the marine collagen for that purpose at about 750 quid a kilo. Now, if as we did, we produce a spun nanofiber from that collagen, and it's used by a company called HRV in a room filter known as the CETA filter. The effective value of the electrospun collagen fiber is 3 million quid a kilo. So if we were able to sell about 20 kilos of that a year, that would equal the value of the hokey catch, the rest of the hokey catch. So I mean, this is a nice thing, because when we think about the technology of fishing, the technology is perfectly there to hoover up virtually every little wriggling creature in the ocean. But if we can have a conversation with the fish cooperatives and say, look, if we do something a bit more interesting and special with your co-product, the stuff that is normally effectively waste, we can improve your profitability, and you can actually cut the catch back so that we do not remove fish from the sea in unsustainable quantities. Now, the reason the cost is so high is because we need very little nanofiber. So each pack of filters that HRV sell to people to clean their rooms of dust and microbes contains about 3 milligrams of material. So that's why the cost is so high. We'll have a look at another example. This was some work with Auckland Medical School. Now, one of the difficulties about making artificial skin is it needs to be thick enough for the surgeon to be able to suture it on to the patient. And so a nice idea is we have a scaffolding, you grow stem cells onto it. The stem cells can come from the patient, and so there's no issue with rejection here. But the trouble is that the cells do not propagate through the thickness of a structural scaffold that's thick enough to suture on at any great speed. In fact, they find it enormously difficult. So this idea is to take a number of electrospun sheets of, it's the same stuff that biodegradable sutures are made out of. So we know fine that the body is quite happy with it. And we produce a number of thin layers where the stem cells grow quite nicely. That's what this mystery item here is showing. It's some confocal microscopy that shows we're getting usable stem cells growing on these ultra thin sheets. Then we stack them all together, and in a very short time they grow into something like this, which is effectively skin made by artificial means that's thick enough for the reconstructive surgeon to suture over a wound in time for it to make some sense as a medical procedure. So we can do lots of things with this. And it's been about a while. Petryanov was a Russian gentleman, and in 1938 he and his colleagues Natalia Rosenblum and Professor Fuchs first of all invented the idea of a nanofiber based filter. Now, people have been having a go at this, but the trouble is with our ultrafine filtering materials that are very intrinsically sticky, and we'll look at that in a bit more depth in a second or two, is they're almost impossible to handle as a thin sheet. It just sticks to everything. So what they decided was, if they took an ordinary filter material substrate and deposited the fibers on top, then they could handle that ordinary substrate and make a filter element out of it, but it would also have many of the desirable properties of an electrospun filter. And here's an example. We can just see a very thin electrospun fiber going down here, and these great big lumps are 60 micron diameter Lycopodium club moss spores that are stuck to it. So quite big things will stick because these fibers have quite a large intrinsic electrostatic charge, so stuff sticks to it. So for a filter, this is great. So there we are, electrospun fibers versus COVID. What's not to like? It's a proven technology. There's large-scale manufacture since 1938. Donaldson in the States do a lot with it. There are one or two other European countries who have electrostatic manufacturers. In the UK, it's the imaginatively named Electrospinning Company, who are preeminent in the field. And it's very effective filtration because we've got the electrostatic attraction, the residual electrostatic field left over from having to cause it to be drawn out by an electrostatic field. That 15 kV means that the filter that we produce has got quite an excess of electrons on it. So a normal filter has to work. Think of a tea strainer. The tea strainer can only work if the mesh on the tea strainer is smaller than the tea leaf. But in this case, as long as our aerosol particulate or virus or whatever hits one of the fibers, it will stick. So in other words, we can have high filtration efficiency and high permeability. So that's lovely. We'll look at the reason why that's important in a second or two. And also, the materials we work with can be biodegradable and sustainable in origin. And you'll recall from the days of the Great Pestilence, the amount of thrown away polypropylene single-use masks. They're neither biodegradable nor sustainable. So what could possibly go wrong? Let's have a look, shall we? So this project, we've called it Future Filter. And our slogan for what's happening is Virus Velcro. So these are, this is why my colleague Fiona Robertson appeared in the co-authorship. Her work was to actually student design and to render it into a form that was stylish and comfortable to wear. And these are some of the free types, and this is the final stone bow mask that she came up with her team. So in common, practically every university, there were people striving to help about the COVID-19 pandemic, and our syndicate was a joint venture between the schools of design, engineering, that's me, and pharmacy to cope with the biochemistry. And what we were wanting to do was to not make, we wanted to make a face covering that offered low resistance to breathing. So it needed to be effective, but it needed not to make you feel as though you had the face hugger from the Alien movies stuck to your face. This is important because think of the difference between a face covering and PPE, personal protective equipment. So a face covering is worn as a voluntary activity. So it's got to be comfortable. Ideally, it should be stylish. And on the plus side, we don't actually need to demonstrate or quantify the efficacy of it. We need to have take a moral position that we believe it works. But for a face covering, it's a voluntary activity. And as long as we can be plausible about it, this is a good situation to be in. If it's personal protective equipment, if we're in the workplace setting, as a manager, you do your risk assessment and say, okay, unfortunately, we're unable to mitigate the risk in any other way but with personal protective equipment. The user, your staff, is absolutely obliged to adopt the recommended PPE. They can't say, I don't like it. Well, they can say they don't like it, but if they're not going to use it, they can be shown the door. And because we are really needing to demonstrate the efficacy of this, there's a whole different raft of testing that needs to be done that would actually show that if you were wearing a mask to protect yourself against the COVID virus, you actually need to demonstrate that there would be an improvement in your safety as a result of using the PPE. So we're in the face covering. The reason being that we lacked at that stage the resources to complete the test regimes that are needed to move us into PPE. Now, when you are thinking of designing, doing some research that needs to go to market, it needs to be done in a concurrent fashion. So the first thing we did is we all rummaged our address books and we put together a supply chain, which is shown here. So Gluco Technologies, who are a polymer development company in Bradford, to supply the polymers we thought we'd need. Our work at the university to make electrospinnable and surface active COVID specific materials. We needed a substrate to put it on. Alstrom Muncio in Finland were one company we approached, and they had a wide range of non-wovens that were sort of suitable. And also WBL Fabrics, again in Bradford, who we eventually went with. We then needed somebody on board to actually make the electrospun fibre. So we approached a number of companies to see whether they were interested. You have to do this at the beginning of the program. There is no point at all in imagining that you could make your little samples and then nip along to a company and say, oh, do you mind making this? You need to get them on board from the beginning, partly so they can feed back and say, actually, if you do it like that, it won't suit us. But also because it's you need to manage the uncertainty in adopting new materials. We had some end users in mind. Arctis Endurance Textiles, based in Ballymena and Exeter, were looking to get into the PPE market, and Countryside Arts in Spilsby, a local company to us. If you buy a commemorative tea towel from your next National Trust visit, probably comes from these people. So their market went a bit quiet during COVID, so they were quite keen to develop something new, and they were very helpful in developing the original concept into something that they could make and sell at a profit. So again, from URL, Owen Phillips was the student who came up with the original concept for the design. Fiona Robertson worked with him and with Countryside Art to produce something that we felt could get into the market. Let's go back to the technical side of it. We'll come to the seaweed in a second. So an effective filter usually has low permeability. If you think about it, you know, if you wanted 100% efficient filter element, how about cling film? Nothing gets through that, so it's 100% efficient. Typically, the filter mesh size needs to be equivalent to the size of the filtrate. You need to catch stuff. It needs to become entangled in your material. My tea strainer example. Now, if you are asked to wear something that is highly efficient and it's got a high resistance to flow, it's going to be difficult to breathe, and you want people to willingly adopt these materials, and if it feels like they're suffocating, they're going to be a bit resistant to it. So the other thing about low permeability is if we think about a mask that is a face covering rather than a hermetic seal, rather than the sort of thing you might wear to resist concentrations of organic vapor or severe dust or something like that, it will clamp to your face. These ones just wrap around your face. So low permeability actually encourages more of the flow of air in and out to go through the filtering element rather than around the edges. So the seaweed. So this is work by Dr. Wright and Prof. Blagden of the School of Pharmacy at the University of Lincoln. First of all, we identified a paper by Kwon et al. In Nature Cell Discovery. So it's a very well thought of publication. And they said, fucoidan, it's sold as a nutraceutical. So it is available. It's extracted from brown seaweeds like bladderwrack. And they noted that for some reason, the COVID virus stuck very well to surfaces that had this material on it. So this is good for us. I mean, we didn't really have any money for testing at this stage. We had a small fund and we bought some fucoidan, and we discovered we could process it. It works fine in terms of being processable, and we'll come to the testing in a minute, but it is a bit whiffy. And then again, we come back to how it would be if we're wanting people to voluntarily use this material. If it smells like, you know, Robin Hood's Bay at low tide in the high summer, there's going to be a little bit of resistance to it. Let's have a look at how it works, though. So what's happening here is there's a chemical mimicry, effectively. So when our COVID virus impacts on a treated surface, we kind of fake it into believing it's hit a lung or a mucosal membrane. So it discharges its messenger RNA and sticks firmly to the surface and is thereby immobilized and rendered inactive. Now, for those of you who are fans of biochemistry, which I regret doesn't really include me, this is why I was delighted to have Dr. Wright and Prof. Blagden on board. They were able to understand the mechanism. So these are actually representations of those spiky bits on the surface of the COVID. And I'm not even going to attempt to explain what's going on, except the protein parts seem to be very well hidden. So it's quite difficult for a vaccine to detect the presence, as it were, or for our immune system to detect the presence. But our biochemistry, the understanding of the biochemistry by our team here, in the first instance enabled us to understand what's going on. We worked with the fucoidan in the first instance, and we thought we had better get some independent laboratory tests done. So we went to Microbiological Solutions Limited in Bury in Lancashire, and we had a few materials tested. The beta-glucan comes from my old colleagues in New Zealand. And so what we can see here, here's our control fabric. So we used a biodegradable cotton felt. And if you try this percent, the test is ISO 18184, and it's the percentage of human coronavirus immobilized on the surface. So if we just take the felt, it's immobilizing about 55%. The New Zealand one actually only immobilized 31.8%. They made the samples and they posted them over to us, and I included them in the test regime. So beta-glucan is extracted from barley, and what we found was the beta-glucan was astonishingly effective. The 30% for untreated felt, 97% retained for treated felt. So that's highly effective. So there we are. Fortunately or unfortunately, my former employees have the right to manufacture this material, so we just pass the results on to them to use as they wished. Our fucoidan, 99% efficient. That's great. And from the understanding developed by Emma and Nick, we were able to try another couple of materials which also were highly effective in specifically retaining the COVID virus on the filter element. Now, go back to our supply chain. You know, for a successful commercializable piece of research work, you jolly well need to have the whole supply chain in place in order to get people on board. If it's just all, you know, and then a miracle occurs, somebody will come along and sell it, people are quite resistant to that. So let's have a look at how this evolved. Our first loss was Arctis Endurance. They realized that the PPE market was absolutely saturated, and they could not really see a way that by the time they'd fronted up the cash for paying for the necessary testing to enter the PPE, that they would be able to make a decent profit of it. My second little difficulty was that all the companies I approached to see whether they could take this up as a manufacturing thing, in principle, yes. In practice, either they were unable to source biodegradable substrates, which we wanted, or they only worked at small scales. So rather unfortunately, all that beautiful electrospinning stuff we were unable to do. And so that cut out the need for a supply of polymers. And actually, Alstrom Munksjö weren't able to supply a biodegradable material either. So it put us in a little bit of a position. So we scrapped off the electrospinning idea. What we actually found was that we could calendar the active material directly onto the substrate, and we constructed the apparatus that is the ISO test for permeability, and we found it didn't affect the permeability of the filtration element. So actually, all that work we did on electrospinning, jolly interesting, but actually not necessary. We could have a process that it was quite possible for our mask manufacturer to adopt in their factory using a locally supplied, well, Brayford to Spilsby, material, a biodegradable cotton felt to produce a face covering. So there we are. The importance of science meeting art. The filter of its own is no use at all. What can we do with it? We need to find somebody who's prepared to make a face covering that would be stylish and usable. And the idea is the face covering itself, this bit you keep and you drop it in the wash once a week or whatever, but every day you put a new felt filter in, and that will protect you during the course of the day. We are slaves of many masters here at the university. It's necessary for us to claim impact for the work we do. It more or less directly leads to extra funding. So we published an article in Materials World, the Journal of the Institution of Materials. It was carefully designed so that it focuses on the fucoidan. You'll notice I've mentioned the beta-glucan, which is a material that we don't have any rights over, and there were a couple there that were only identified by letters. That's our patentable intellectual property. So the way it works, for those of you who are not familiar with the wacky world of academic success, is you must have a publication to indicate that you have done the work, and then you need to get somebody to take it up and use it. So this is our publication. We may yet do more working on that. And in further developments are other novel active filter materials that are very secret, are in the process of being patented by the university, and the university proposes to license this to various end users to get it out into the market. I must admit, my original thought was we'll just tell people and they can use it. But the commercial advisory team of the university said, actually, that's a terrible way of getting people to adopt it. You will do far better to license it to a manufacturing company. So that's what's happening at the moment. With any of this, you know, I'm only giving you the 1/10 of the visible iceberg. So the companies that have been involved and the institutions I would like to acknowledge. And also, when we look at the enormous team of people who over the years have been helpful to put us in the position that we could move rapidly to produce exploitable science and technology, these are they. So I now thank you for your kind attention, and I hope you found that of interest.